Imagine your brain right now. Just picture it as a sprawling, high-stakes metropolis that simply never sleeps.
A city running on absolute overdrive, yeah.
Exactly. And the streets of this city are absolutely flooded with this explosive, highly volatile accelerator fluid. At any given second, a single failure in the containment systems could just, well, it could cause the entire city to burn itself to the ground.
It is a terrifying thought when you really break down the biology of it.
Right. But keeping that city from catching fire while simultaneously, harnessing that explosive energy to drive every thought, every single memory, and every movement you make, it is a microscopic miracle happening millions of times a second.
Spot on. And that delicate balance is exactly what we are unpacking today.
Because, as we always say here, psychiatry is the study of how brain, mind, and experience interact and how that process can go wrong. That is a profound anchor for us to hold on to as we embark on today's deep dive.
It really is the perfect framing for what we are tackling today. If you look at the history of mental health, and particularly the biological side of psychiatry, there's been this glaring asymmetry in what we actually pay attention to.
What do you mean by asymmetry? Like, we've been looking at the wrong things.
Well, not the wrong things, but certainly not the whole picture. For about 70 years, the entire field has been obsessively focused on a tiny group of chemicals known as the biogenic amines.
Ah, right. The celebrities of the brain. We're talking about dopamine, serotonin, norepinephrine.
The exact ones.
Yeah.
They get all the press, don't they? They are the targets of almost every classic psychiatric medication, from SSRIs to your standard antipsychotics.
Yeah, I mean, if you ask anyone on the street about depression, they'll immediately say serotonin. But today, we are sort of pushing those celebrities aside, aren't we, to look at the actual heavyweights?
We are indeed. The amino acid neurotransmitters, primarily glutamate and GABA. And honestly, the sheer scale of the shift in perspective this requires, it cannot be overstated.
Really? Is it that massive of a difference?
Oh, absolutely. Those famous biogenic amines, dopamine and serotonin, they exist in the brain in micromolar concentration.
Micromolar, so tiny amounts.
Exactly. They are basically trace elements. They act as these distant, sort of broad modulators of the system. But the amino acid neurotransmitters, glutamate and GABA, they are present in millimolar concentrations.
Wait, millimolar? So that's...
Thousands of times more abundant.
That's right.
Yes, in fact, they are so heavily concentrated that we can actually measure them in a living human brain using magnetic resonance spectroscopy.
That's the MRS scan, right?
Right. And you can do it without exposing the patient to any radiation whatsoever. You can literally see the peaks of glutamate and GABA because there is just so much of it.
That is wild.
It is. Virtually every single neuron in the central nervous system is innervated by glutamatergic and GABAergic projections. So they are not just modulating the architecture of thought. They literally are the foundational architecture.
So if we go back to our city metaphor for a second, glutamate is that explosive accelerator fluid I was talking about. It drives the traffic forward. It is the engine of excitation, of signal transmission, basically, of waking up the city.
Yes, the pure excitatory drive.
And GABA, on the other hand, is the intricate system of brakes, the traffic lights, the thick concrete barriers. It is the inhibition that keeps the city from spinning into absolute gridlocked chaos.
That is a brilliant way to conceptualize it. And every psychiatric symptom a person experiences, essentially, represents a shift in this very delicate tone.
So too much excitation and the city burns. You get excitotoxicity, seizures, instability.
Precisely. And conversely, too much inhibition, the city just, well, it shuts down entirely. You drift into suppression or even a coma.
Which brings us to our mission for this session. We're going to build a robust conceptual synthesis of this dynamic negotiation, moving from the microscopic mechanisms at the synaptic cleft all the way up to the clinical manifestations of psychiatric disease.
And we are using Kaplan and Sadok's comprehensive textbook of psychiatry as our structural spine today, specifically Chapter 1.6.
But just to be clear to you listening, we are not just cataloging dry biochemical facts here. We are exploring the physiological basis of how we learn, how we process trauma, and how the modern understanding of psychiatric pathology is undergoing a radical paradigm shift.
The shift away from those simple chemical imbalances we used to talk about and towards complex circuit tuning.
Right. So let's dive in. If glutamate is the primary accelerator driving all this, just how much of the brain's real estate does it actually take up?
Well, glutamate mediates fast excitatory neurotransmission, and it is responsible for approximately 80% of all brain synapses.
80%.
Yes, particularly those that innervate dendritic spines. and the energy demands of the system are frankly staggering.
I would imagine if it's doing 80% of the work.
Exactly. When a glutamatergic signal fires, it depolarizes the neuronal membrane. The simple act of repolarizing those membranes just to prepare for the next signal.
Just to reset the system.
Right, just the reset accounts for up to 80% of the brain's total energy expenditure. It is the highest concentration of any amino acid in the brain, sitting at about 10 millimolar.
And is all of that active? Like ready to fire?
Not all of it. About 20% of that represents the active neurotransmitter pool that is sitting there waiting to be released.
Okay, so if it is that abundant and that critical for energy, my first thought is, well, the brain must just pull it straight from the food we eat, right? Like from dietary protein.
You would think so, but the blood-brain barrier actually shuts it out completely.
Wait, why would the brain block its own primary fuel source? That sounds counterintuitive.
Because it is simply too dangerous. Glutamate is highly excitatory, and as we will trace later on, it is potentially neurotoxic in high ambient amounts.
Ah, right, the explosive fluid.
Exactly.
Yeah.
If the brain allowed dietary glutamate to just cross the blood-brain barrier willy-nilly, every time you ate a meal rich in amino acids, you would risk profound excitability, or even seizures.
Oh, wow. So a steak could literally cause a seizure?
Theoretically, yes, if the barrier wasn't there. The brain has to strictly control its own supply. Therefore, it synthesizes glutamate de novo from scratch, from glucose, entirely within its own borders.
Walk us through that manufacturing process then. How on earth does a simple sugar become an explosive neurotransmitter?
It connects directly to fundamental cellular metabolism. The brain uses the tricarboxylic acid cycle.
The TCA cycle, or the Krebs cycle, as I remember it from uni.
That's the one. It uses the Krebs cycle to generate a molecule called alpha-ketoglutarate. Then, through a transaminase reaction, an amino group is added to that alpha-ketoglutarate, which converts it into glutamic acid.
So it's basically a highly efficient closed-loop system.
Very efficient. Virtually all the glucose that enters the brain cycles through glutamic acid at some point. And the portion that is destined for neurotransmission is then actively sequestered into synaptic storage vesicles.
By a transporter, I'm assuming.
Yes, by a specific transporter called the vesicular glutamate transporter, or VGluTT. It packs the glutamate into these vesicles, where it just waits for an action potential.
Okay, so let me picture this. An electrical signal travels down the neuron. The vesicle fuses with the membrane, and it dumps this high-energy glutamate right into the synaptic cleft to activate the next neuron.
Correct.
But then it has to be cleared away instantly, right? Or the system just burns out from overexcitation.
Exactly.
Now, logically, I would assume the neuron that just released it simply pulls it back in, recycles it, and puts it straight back into a vesicle. But I was reading the sources, and it indicates the nerve terminal releasing the signal doesn't even possess the equipment to clean up its own mess.
That is true.
That seems incredibly inefficient. Why would it do that?
It does seem that way, but it forces us to completely redefine what a synapse actually is. Historically, neurobiology taught a two-way street model. A presynaptic terminal releasing a chemical and a postsynaptic terminal receiving it.
Right, the classic diagram in every textbook.
Yes. But glutamate demands we adopt the model of the tripartite synapse.
Tripartite, so 3 parts. Who is the third partner in this negotiation?
The third, absolutely indispensable partner is the astrocyte, which is a type of glial cell.
Okay, so what is the astrocyte actually doing?
Astrocytic end feet intimately envelop and wrap around the entire glutamatergic synapse. And it is the astrocyte, not the glutamatergic nerve terminal, that expresses the crucial excitatory amino acid transporters, specifically EAT1 and EAT2.
So the astrocyte acts like a hazardous materials team.
Yes, a hazmat team.
The neuron releases the explosive material, and the astrocyte rushes in with these EA2 transporters to hoover it up and secure the area.
That is a perfect analogy.
But what do they do with it once they hoover it up? Because they can't just hand the raw explosive material back to the neuron for the next signal, can they?
They absolutely do not. Once the glutamate is safely inside the astrocyte, an enzyme called glutamine synthetase takes over. And this is a critical detail for you to remember. Glutamine synthetase is an ATP-dependent enzyme that is exclusive to glial cells.
So it's not in the neurons at all?
Not at all. It is only in the glia. It catalyzes the conversion of the highly excitatory glutamate into glutamine.
And glutamine is safe?
Yes, glutamine is a safe, inert, neutral molecule.
Okay, so the hazmat team packs the explosive material into a stable, padded box.
That is a highly accurate way to visualize it. This stable box glutamine is then released by the astrocyte back into the extracellular space.
And then the neuron can finally pick it up.
Exactly. It is finally taken back up by the original glutamatergic neuron. And once inside the neuron, a mitochondrial enzyme called phosphate-activated glutamase unpacks the box.
Converting the inert glutamine back into the active glutamate.
Precisely. Ready to be put into a vesicle all over again. This elegant loop is known as the glutamine cycle, and it accounts for about 40% of all brain glutamate turnover.
If you're listening to this and wondering why on earth this microscopic supply chain matters clinically, just think about what happens if that astrocytic hazmat team fails.
It is catastrophic.
Right. If their equipment breaks down, the neuron is completely helpless. The explosive material just builds up in the street.
And that is not just a hypothetical scenario. If the EAT2 transporters on the astrocyte fail, extracellular glutamate levels rise rapidly. This leads straight to excitotoxic cell death.
Do we see that in actual patients?
We do. We observe this specific loss of EAT2 protein and transport activity in the ventral horn of patients with amyotrophic lateral sclerosis, or ALS.
Oh, wow. So ALS involves a failure of this exact cleanup system.
Yes. The tripartite synapse is quite literally a vital life support system. Without the astrocyte constantly sweeping the cleft, the neurons would quickly destroy themselves.
All right, I think we have a solid grasp on how the accelerator fluid is made and managed. Let's look at the engines that actually revs up. When glutamate crosses the synaptic cleft, what physical locks is it turning?
The postsynaptic effects are mediated by two major families of receptors. First, we have the metabotropic glutamate receptors, or MGLRs.
Metabotropic, so they involve metabolism or secondary messengers.
Exactly. These are G-protein-coupled receptors, somewhat similar to how dopamine receptors work. They do not open ion channels directly. Instead, they trigger complex intracellular signaling cascades that fine-tune the local environment. They are modulators.
Okay, so they're the slow adjusting dials. What about the fast ones?
The structures responsible for fast, instantaneous excitatory neurotransmission are the ionotropic receptors, the ligand-gated cation channels. And these are divided into three main families: AMPA, kinetic, and NMDA.
Let's start with AMPA, because they seem to be the reliable, everyday workhorses of the brain. You want to move a muscle or process a simple thought, you use an AMPA receptor.
Yes, AMPA receptors mediate the fast, depolarizing postsynaptic currents. When glutamate binds to an MPA receptor, a channel physically pops open, allowing sodium ions to rush into the cell.
And sodium carries a positive charge.
Right. Because it carries a positive charge, this rapidly changes the electrical voltage of the membrane, which propagates the signal. But, and this is fascinating, even these everyday workhorses possess astonishing complexity, particularly regarding their permeability to calcium.
Yeah, I wanted to ask about this. The textbook mentions something called QR editing, controlling that calcium permeability. And I have to admit, the idea of the brain literally editing its own instructions on the fly sounds a bit like science fiction. What does that actually mean?
It is one of the most remarkable regulatory mechanisms in neurobiology. So the AMPA receptor is built from multiple subunits, right? One of which is called GLOR2.
Okay, GLOR2.
The DNA gene for GLOR2 encodes for an amino acid called glutamine, which is represented by the letter Q, a very specific position inside the channel pore.
So the default instruction is to put a Q there.
Exactly. However, after the DNA is transcribed into messenger RNA, but before that RNA is translated into a protein.
Wait, so while the blueprint is in transit...
Yes. While it's in transit, an enzyme called adenosine deaminase intervenes. It physically alters the messenger RNA sequence. It converts the codon for glutamine, the Q into the codon, for a completely different amino acid, arginine, which is represented by R.
Wait, hold on. You're saying the brain reads the genetic blueprint and then actively crosses out a word and writes in a different one before handing it to the builders.
That is exactly what happens.
That is insane. What does that actually achieve physically?
The structural consequence is profound. You see, glutamine is a neutral molecule, but arginine carries a Berkey positive charge.
And positive repels positive.
Spot on. Calcium ions are also positively charged. So if the receptor subunit incorporates that bulky, positively charged arginine, it acts as a physical and electrical barrier to calcium ions. It only lets sodium through.
Ah, I see.
Most AMPA receptors in the adult brain are fully edited this way. But if the editing process is incomplete and the glutamine remains, the channel pore is wider and lacks that positive repelling force.
Meaning calcium can just flood right in.
Exactly. The channel suddenly becomes highly permeable to calcium, and this completely alters the postsynaptic response because calcium acts as a powerful intracellular trigger. The brain dynamically tunes the physical properties of its own hardware by actively rewriting the RNA.
That is just staggering. And then alongside AMPA, we have the kainate receptors. From what I gather, they operate a bit differently, often sitting on the presynaptic side.
Kainate receptors are indeed less involved in fast postsynaptic signaling and much more involved in local circuit regulation. They are frequently situated on the presynaptic terminals themselves.
So they act like sensors for the neuron that's doing the releasing.
Yes, they act as autoreceptors. When glutamate concentrations in the synaptic cleft rise high enough, the glutamate binds to these kainate autoreceptors, which then act to throttle back further glutamate release. They serve as a local presynaptic negative feedback loop.
A built-in safety valve.
Exactly.
But the absolute crown jewel of this system, the receptor that redefines how we understand memory, learning, and major psychiatric disease, is the NMDA receptor.
The NMDA receptor is a marvel of biophysics, truly. It possesses features that make it entirely unique among all neurotransmitter receptors in the human body. It functions as a biological coincidence detector.
A coincidence detector. I always picture it like the launch console in a nuclear submarine. You can't just press one button. You need multiple keys turned simultaneously by different people in different places just to open the vault. Walk us through the specific keys required here.
It is a great analogy. To open the NMDA receptor channel, 3 distinct conditions must be met concurrently. First, you need the primary chemical key. Glutamate must bind the NR2 subunit of the receptor.
But unlike AMPA, just having glutamate binding does nothing on its own.
Correct. It is insufficient. Second, you require a coagonist key. A completely different molecule must bind to what is known as the glycine modulatory site, or GMS, which is located on the NR1 subunit.
Okay, so we have 2 chemical keys in the locks. But even if you have both of those firmly in place, the door still refuses to open, right?
Because of the third, biophysical lock. Wedged tightly inside the central pore of the NMDA channel is a large magnesium ion.
Just physically blocking the hole.
Yes. At a normal resting electrical membrane potential, that magnesium plug sits there, completely immovable. The receptor remains absolutely silent and deaf to the glutamate binding to it.
So how do you get rid of the plug?
The only force capable of removing that magnesium plug is voltage. The surrounding neuronal membrane must become heavily depolarized, meaning it becomes highly positively charged inside.
And how does it get that positive charge?
Through the rapid simultaneous firing of the local AMPA receptors we just discussed.
So the everyday workhorses have to do the heavy lifting first.
Precisely. So for the NMDA vault to open, you need presynaptic glutamate release, you need the coagonist present at the GMS, and you need intense converging AMPA activation to depolarize the membrane.
And when the inside of the cell gets positive enough?
It electrically repels the positively charged magnesium ion, forcefully ejecting it from the pore.
It requires an intense consensus of signals. The whole local neighborhood of the neuron has to be screaming, "This is an important event, pay attention," before the NMDA receptor finally activates. And when it does open, what rushes in?
Massive amounts of calcium. Unlike the sodium influx of AMPA, which just transmits a quick electrical spark, the calcium influx through the NMDA receptor acts as a second messenger.
Meaning it changes things inside the cell.
Yes. Calcium binds to internal proteins, activating various kinases, like calmodulin activated protein kinase 2. These enzymes then physically travel to the nucleus and alter the actual gene expression of the neuron.
Wow.
The NMDA receptor is literally the mechanism by which the brain translates temporary electrical experiences into permanent structural changes.
Let's linger on that second key for a moment, the coagonist at the glycine modulatory site, because you'd assume it's just glycine, given the name.
That assumption held for many years. And in the brainstem and spinal cord, glycine is indeed the primary coagonist. But in the forebrain, the corticolimbic regions responsible for higher-level cognition, emotion, and complex psychiatric states, the dominant coagonist is actually D-serine.
Which is strange, isn't it? Because from basic biology, I thought mammals only utilized L-amino acids. I remember learning that D-amino acids were supposed to be something only found in bacterial cell walls.
It was long considered a biological dogma that D-amino acids had no physiological role in mammals. But the human brain intentionally breaks this rule.
How does it even get the D-serine?
It uses an enzyme called serine racemase to deliberately convert L-serine into D-serine, specifically to prime these NMDA receptors. Furthermore, the synthesis and release of this D-serine are heavily regulated by the astrocytes.
The hazmat team again.
Exactly. The hazmat team is also actively managing the keys to the nuclear vault.
It paints such a vibrant picture of the synapse, but these receptors aren't just floating freely like rafts on a cellular ocean, are they? The textbook refers to them being anchored to an osmophilic multi-protein complex, which, let's be honest, sounds completely impenetrable. What does that actually look like in conversational terms?
Think of the postsynaptic density, the PSD, as a massive, dense scaffolding of proteins poured right at the receiving end of the synapse. It is essentially the concrete and steel foundation that holds all these receptor folds in place.
Okay, a steel framework.
The principal organizing protein in this foundation is called PSD95. The NMDA receptors bind directly and rigidly to specific slots called PDZ domains on PSD95. They're bolted down securely.
So they don't move.
No. And this steel framework also holds effector enzymes, like nitric oxide synthase, right next to the NMDA receptor pore. So the moment calcium enters, the enzymes can react instantly.
But the AMPA receptors, the everyday workhorses, they aren't bolted down the same way, are they?
They are not. AMPA receptors do not bind directly to PSD95. They attach loosely via intermediary proteins such as TARPs, which stands for transmembrane, AMPA receptor regulatory proteins, and GRIP.
Why the looser connection?
It is a brilliant design feature because it allows AMPA receptors to be highly mobile. While the NMDA vault stays fixed, AMPA receptors can be rapidly shuttled in and out of the synapse like modular furniture.
Oh, I like that, modular furniture.
In fact, early in development or in certain states of memory formation, there are synapses that possess bolted-down NMDA receptors but absolutely zero AMPA receptors.
So wait, if NMDA receptors require AMPA receptors to depolarize the membrane and kick out the magnesium plug, a synapse with no AMPA receptors is completely useless, isn't it? It's totally deaf to resting signals.
It is functionally silent. We literally call them silent synapses. They exist structurally, but they transmit no baseline electrical signal. They're just waiting to be recruited. And that mechanism of recruitment brings us directly into how the brain actually learns.
Let's talk about that, because this is the core of it. How do these silent synapses wake up? How does this intricate lock system translate to me remembering where I parked my car or learning a new language?
We are entering the realm of Hebbian plasticity, the famous neurological principle that, quote, neurons that fire together wire together. In the 1970s, neurophysiologists discovered that if they applied a brief, intense burst of high-frequency electrical stimulation to a glutamatergic synapse in the hippocampus, the subsequent and normal signals set across that synapse were suddenly much stronger.
And they stayed stronger for a long time.
Days or weeks. They term this long-term potentiation, or LTP.
And I assume the reverse is true. If they stimulated it with a slow, low-frequency pulse, the connection permanently weakened, which they called long-term depression, or LTD.
Correct. And the master switch for this entire phenomenon is the NMDA receptor. If you block the NMDA receptor with a pharmacological agent, you completely abolish the brain's ability to undergo LTP. You literally turn off the ability to record new long-term memories.
So what physically changes in the synapse during LTP to make the connection stronger? Is the neuron just releasing more glutamate?
The primary change happens postsynaptically, relying on that modular furniture concept we discussed. The massive influx of calcium through the activated NMDA vault triggers an intracellular cascade. This cascade physically shuttles reserve AMPA receptors from inside the cell and inserts them right into the postsynaptic density.
Oh, so those silent synapses are suddenly populated with AMPA receptors.
Yes. The synapse essentially builds a wider lane for traffic. The more AMPA receptors present on the surface, the stronger the depolarizing current will be the next time a normal amount of glutamate is released.
But it goes beyond just adding receptors, doesn't it? Because using modern imaging techniques, we can actually see the physical shape of the brain changing.
Yes, using two-photon confocal microscopy, researchers have literally watched dendritic spines change shape in real time following LTP. Before heavy NMDA activation, a dendritic spine might look like a long, skinny, immature appendage. Following the calcium influx and the insertion of AMPA receptors, that spine physically balloons outward. It transforms into a fat, stubby, mature mushroom shape. This massive increase in surface area creates a much more robust and permanent synaptic contact.
But expanding a physical structure requires raw materials. Where's the neuron get the building blocks to inflate that spine so quickly? It can't wait for a shipment from the main cell body, can it?
No, it manufactures them right there on site. This structural plasticity requires the local synthesis of new proteins directly within the dendritic spine, bypassing the need to send a request all the way back to the cell nucleus.
Local manufacturing, how is that managed?
It is regulated by specific RNA-binding proteins, the most notable being Fragile X mental retardation protein, or FMRP, FMRP chaperones specific messenger RNAs to the spine and oversees their translation into structural proteins.
Which provides a devastatingly clear explanation for Fragile X syndrome.
Precisely. If the gene encoding FMRP is mutated and non-functional, the local protein manufacturing plant is broken. When you look at the brain of a patient with Fragile X syndrome, you see an abnormal overabundance of thin, spindly, immature dendritic spines.
Because they can't make that mushroom shape.
Right. They cannot physically lock in the structural changes required for long-term learning, which leads to severe intellectual disability and neurobehavioral deficits.
This understanding of plasticity isn't just about pathology, though, is it? If NMDA is the master switch for learning and it operates on these structural levels, can we manipulate it therapeutically? Like, think about a patient with severe PTSD or a debilitating phobia. Their brain has effectively learned a pathological, overpowering fear response to a specific trigger. Can we hack the NMDA receptor to help them unlearn that trauma?
That is one of the most exciting translational applications in modern psychiatry. The process of overcoming a phobia called fear extinction is not simply forgetting. It is an active biological process of learning a new, safe association that overrides the old fear network.
And learning requires NMDA.
Exactly. Just like any learning, fear extinction absolutely requires NMDA receptor activation, particularly in the amygdala.
So we need to make those NMDA receptors more sensitive during a therapy session. How do we do that safely?
Researchers have utilized a drug called D-cycloserine. Historically, D-cycloserine is an old antibiotic used to treat tuberculosis. But, quite coincidentally, it happens to act as a partial agonist at the glycine modulatory site on the NMDA receptor.
It's a pharmacological master key that slightly props open that second lock on the vault we talked about.
Exactly. In landmark clinical trials, patients with severe acrophobia and intense fear of heights were given a single dose of D-cycloserine shortly before engaging in a session of virtual reality cognitive behavioral therapy.
And what happened?
The D-cycloserine robustly enhanced the efficacy of the psychological intervention. By chemically priming the NMDA receptors for plasticity, the patients were able to acquire the new fear-extinguished learning much more rapidly, and the clinical improvements persisted for months compared to the placebo group.
That is brilliant.
We are using targeted pharmacology not to sedate or numb symptoms, but to actively bolster the brain's neuroplastic capacity for psychological healing.
It is a beautiful synergy of psychotherapy and neurobiology, but we need to look at the dark side of this mechanism, because if the accelerator gets stuck, if the NMDA vault stays open too long, the calcium doesn't trigger learning. It triggers death. Excitotoxicity.
Yes. If we return to our city metropolis metaphor, excitotoxicity is what happens when the traffic systems fail entirely. The vehicles accelerate uncontrollably, and they just crash straight through the building.
A total system failure.
If glutamate accumulates in the synaptic cleft and persistently, overwhelmingly activates AMPA and NMDA receptors, the neuron loses all ability to maintain its internal environment. You get a massive, unregulated influx of sodium and calcium.
And in biology, water always follows salt.
Yes. The overwhelming influx of sodium draws water into the cell via osmosis. The neuron rapidly swells, suffers acute cellular edema, and eventually bursts. This is rapid necrotic cell death.
But the calcium causes a different kind of death, doesn't it?
It does. It is a second, more insidious mechanism driven by the calcium. Even if the cell doesn't burst immediately, a persistent elevation of intracellular calcium profoundly disrupts the mitochondria. The struggling mitochondria release cytochrome C, which acts as an internal self-destruct signal.
Activating those enzymes, what are they called?
Caspases. It activates a cascade of caspases, which systematically dismantle the neuron's DNA and cytoskeleton from the inside out. This is programmed cell death, or apoptosis.
Let's apply this to a clinical emergency just to make it concrete. If someone is having an ischemic stroke, a blood clot blocking flow to the brain, we usually say they are dying from a lack of oxygen, but based on what you're saying, the lack of oxygen is just the match. The brain actually poisons itself with its own accelerator fluid. Trace that step by step for us.
It is a devastating cascade. When the blood clot hits, local tissue hypoxia occurs. Without oxygen, the mitochondria instantly stop producing ATP.
The energy currency.
Right. Without ATP, the energy-hungry sodium-potassium pumps on the cell membranes fail. The strict sodium gradient that normally exists across neuronal and glial membranes collapses entirely.
And remember, our astrocytic hazmat team from earlier. Their glutamate transporters, EAT1 and 2, completely rely on that strict sodium gradient to pull the explosive glutamate off the streets.
That is the critical failure point. When the sodium gradient collapses, the physical transporter mechanism of the EATs actually reverses direction.
They reverse.
Yes. Driven by the collapsing gradient, the astrocytes literally vomit massive amounts of stored glutamate back out into the synaptic cleft.
Oh, that's horrific.
It is. The local NMDA receptors are blown wide open, calcium floods in, and a catastrophic wave of excitotoxic cell death ripples outward from the core of the stroke.
It's terrifying. But excitotoxicity isn't just an acute explosion like a stroke, is it? The literature points to this happening in slow motion over decades, in conditions like Alzheimer's disease.
In Alzheimer's, the pathology is subtle but relentless. The hallmark beta-amyloid fibrils, specifically beta-amyloid 142, orchestrate a multifaceted assault on the glutamate system. The fibrils chronically, slightly depolarize the neuronal membranes. This constant mild depolarization is just enough to chronically dislodge that protective magnesium block from the NMDA receptors. Furthermore, the amyloid impairs the glutamate transporters on the astrocytes, meaning ambient glutamate lingers slightly longer in the synapse.
So the vault doors are basically slightly ajar all the time, constantly leaking toxic levels of calcium into the cell. If Alzheimer's is basically a slow-motion excitotoxic leak, why can't we just give the patient a drug that permanently blocks the NMDA receptors, lock the vault entirely to save the brain?
Well, because if you fully block the NMDA receptors, you completely abolish the patient's ability to form any new memories at all. You would induce a state resembling profound dementia or acute psychosis.
Right, which defeats the purpose.
Exactly. This is why the drug memantine is such a clever pharmacological intervention. Memantine, an FDA-approved treatment for moderate to severe Alzheimer's, is a low-affinity, uncompetitive antagonist of the NMDA receptor.
It acts like an artificial, slightly modified magnesium plug.
Exactly. Because it has a relatively low affinity, it gently plugs the channel just enough to stop the continuous low-level toxic leak of background calcium.
But it doesn't block the real signals.
No. Because it doesn't bind too tightly, when a massive physiological burst of glutamate arrives to form a real memory, the memantine is easily knocked out of the way, allowing normal phasic signaling to occur. It filters out the toxic noise, but lets the vital signal through.
Okay, so if we have this incredibly dangerous explosive accelerator constantly firing, what's stopping our brains from just burning out in continuous seizure? There must be an equally powerful braking system.
Right.
Which brings us to the restraint, the calm, the traffic lights of our neuronal metropolis. GABA.
Gamma-aminobutyric acid, or GABA.
Yeah.
It is the major inhibitory neurotransmitter in the central nervous system. And yet, its very existence represents a profound biochemical irony.
The irony being that the brain takes the most explosive substance it has, glutamic acid, and snips off exactly one chemical bond to turn it into the ultimate fire blanket. That sounds like playing with fire. Why would the brain build its brakes out of its accelerator fluid?
It speaks to the brutal efficiency of evolutionary biology. Instead of maintaining two entirely separate energy-intensive synthetic pathways, the brain utilizes a single, readily available precursor.
So how does it convert it?
The conversion is catalyzed by an enzyme called glutamic acid decarboxylase, or GAD.
Yeah.
D removes a single carboxyl group from glutamate, instantly transforming its physiological function from purely excitatory to purely inhibitory.
And where does this manufacturing happen? Is it in the same cells?
No, GAD is exclusively expressed within GABAergic neurons. Interestingly, there are two distinct genes that encode it. GAD65 is localized primarily to the nerve terminals, and it synthesizes the rapid response pool of GABA destined for immediate vesicular release.
And the other one?
GAD67. It is distributed evenly throughout the cytoplasm of the cell and is responsible for maintaining the baseline neuronal supply. If you breed a mouse genetically lacking GAD67, it is born with profoundly depleted brain GABA levels and dies almost immediately.
And once GABA is released and applies the brakes, how is it cleared away? Does it use the same astrocytic hazmat team?
The clearance relies on high-affinity sodium-driven GABA transporters denoted as GATs. These are located both on the presynaptic terminal itself and on surrounding astrocytes. Once recovered, GABA is catabolized, broken down by a mitochondrial enzyme called GABA transaminase, or GABA-T.
Converting it into what?
This converts GABA into succinic semialdehyde, which eventually finds its way back into the Krebs cycle.
Now, if you're a clinician listening to this, you are intimately familiar with manipulating these clearance mechanisms. Because if a patient arrives in status epilepticus, a continuous life-threatening seizure, their brain is experiencing runaway excitation. You need to artificially increase the braking power immediately. So you might administer a drug like valproic acid, which inhibits GABA-T.
Yes. By inhibiting the enzyme that destroys GABA, you force GABA levels in the brain to rise, enhancing the overall inhibitory tone.
Or you could block the transporters entirely.
Right. You could use an anticonvulsant like tiagabine, which physically blocks the GAD transporters. This leaves naturally released GABA lingering in the synaptic cleft for much longer, prolonging the braking effect. Both pharmacological strategies achieve the same goal, shifting the tone away from excitotoxicity and towards stability.
Let's talk about the physical anatomy of this braking system. If glutamate accounts for 80% of synapses, GABA must be massively outnumbered. How does a minority population of neurons exert enough control to stop the entire cortex from seizing?
It is entirely about strategic placement. In the cerebral cortex, GABAergic neurons are primarily local circuit interneurons. Unlike the massive glutamatergic pyramidal cells that project their axons deep into the spinal cord or across the brain, GABA interneurons act locally. They only account for about 15 to 25% of cortical neurons.
So they are outnumbered 4-to-1.
They are. But consider a specific type of GABAergic interneuron known as a chandelier cell.
Chandelier cell.
They don't waste their energy synapsing on the distal, faraway branches of a pyramidal cell's dendrites. Instead, they place their inhibitory synapses directly onto the axon hillock of the glutamatergic cell.
The axon hillock being
The final bottleneck, the exact trigger point where the pyramidal cell decides whether or not to fire an action potential.
they bypass the negotiations entirely and just cut the main power line.
Precisely. By placing their inhibitory synapses right at the critical bottleneck, a single GABAergic interneuron can effortlessly veto the combined excitatory signals arriving from thousands of distal glutamatergic inputs. They dictate the output of the cortex through architectural supremacy.
Okay, let's examine the actual locks that GABA turns, the receptors. Just like glutamate, GABA has fast ion channels and slow G protein modulators. Let's tackle the fast ones first. GABA.
The GABA receptor is a ligand-gated ion channel, structurally classified as a heteropentamer. It is constructed from 5 protein subunits arranged in a circle, forming a central pore.
So when GABA binds, the pore opens.
Yes, when GABA molecules bind to specific interfaces between the alpha and beta subunits, the channel undergoes a conformational shift and opens. But unlike AMBA or NMDA, which let positive ions in, the GABA re-channel is selectively permeable to chloride ions.
And chloride carries a negative charge.
Indeed. In a mature adult neuron, the concentration of chloride is much higher outside the cell than inside. So when the GABA-A channel opens, negatively charged chloride rushes down its concentration gradient into the cell.
And that extra negative charge pulls the voltage down.
Exactly. This influx of negative charge hyperpolarizes the membrane. It drives the electrical voltage further away from the threshold needed to fire an action potential. It physically makes it harder for the neuron to fire. It slams on the brakes.
But the textbook mentions a fascinating and clinically vital exception to this rule regarding newborns.
It is a profound developmental paradox. In the immature neonatal brain, the expression of chloride transporters is different. The developing neurons actually actively pump chloride into the cell, creating an unusually high intracellular concentration.
Which means the gradient is reversed.
Exactly. So when GABA opens the door in a newborn brain, chloride rushes out of the cell, taking its negative charge with it. This loss of negative charge actually depolarizes and excites the neonatal neuron. Early in development, GABA functions as an excitatory neurotransmitter.
Which perfectly explains why, if a neonate is seizing, giving them a standard anticonvulsant that enhances GABA-like phenobarbital can sometimes paradoxically worsen the seizure. Their brakes are literally wired as accelerators.
It's critical clinical nuance.
But returning to the adult brain, the GABA receptor is arguably the most famous receptor in psychiatry because it is the target of benzodiazepines and barbiturates.
They're among the most heavily prescribed neuroactive compounds in medical history. Let's delineate how they interact with the receptor, because they do so in fundamentally different ways.
Let's use an analogy here. Imagine the GABA receptor is a bouncer working the door at an exclusive nightclub. The bouncer's only job is to let the chloride ions into the club. GABA is the bouncer. When GABA shows up, he opens the door. What are the benzodiazepines like Valium or Xanax doing?
Benzodiazepines bind to a distinct, highly specific allosteric site on the receptor complex, entirely separate from where GABA binds. They cannot open the channel on their own. In your analogy, the benzodiazepine is the club manager standing behind the bouncer, whispering in his ear, encouraging him to be more active.
So they don't open it themselves, they just urge the bouncer to do it more often.
Right. When GABA is naturally released, the presence of the benzodiazepine increases the frequency with which the channel opens. The door opens and closes much faster, letting more chloride in over time.
And because the manager can't open the door himself, he absolutely relies on the bouncer being there. Benzodiazepines have a built-in physiological ceiling. Even if you take a massive dose of Valium, your brain's inhibition is limited by the amount of natural GABA available. This is why benzodiazepines, taken alone, rarely cause fatal respiratory suppression. But what about the older drugs, the barbiturates, like phenobarbital?
Barbiturates are a different class of hazard entirely. They bind to their own distinct site on the receptor. Instead of making the door open more frequently, they alter the channel dynamics by markedly increasing the duration of the open state. They tell the bouncer to prop the door wide open and walk away.
The barbiturate is the owner of the club.
A very dangerous owner. Because crucially, at high enough concentrations, barbiturates lose their reliance on GABA entirely. They can directly open the chloride channel themselves, even if the bouncer is absent.
Oh, that's bad.
Because there is no ceiling effect. Taking an overdose of a barbiturate will drive the inhibition so deep that the autonomous respiratory centers in the brainstem simply stop firing. The patient stops breathing. This direct channel opening mechanism is why barbiturates have such a notoriously narrow therapeutic index and are highly lethal in overdoses.
The complexity of the GABA receptor goes even deeper than that, right? You mentioned it's a pentamer made of different subunits, and the specific recipe of subunits changes what the drug actually does to the patient.
The receptor is typically composed of a combination of alpha, beta, and gamma subunits, and there are multiple variants of each, alpha 1 through alpha 6, for instance. Through sophisticated mutational studies in mice, we have mapped specific clinical effects to specific subunits.
Give us an example.
If a benzodiazepine binds to a GABA receptor containing the alpha-1 subunit, it heavily mediates sedation and anterograde amnesia. If it binds to a receptor containing the alpha-2 subunit, it mediates anxiolytic anti-anxiety effects with significantly less sedation.
This directly explains the design of modern sleep aids. Drugs like zolpidem or Ambien are chemically engineered to bind almost exclusively to the alpha-1 subunit. They intentionally trigger the sedation pathway while attempting to leave the rest of the GABA system alone. We also have to discuss neurosteroids, because this represents one of the most significant paradigm shifts in modern psychopharmacology.
For a long time, we viewed hormones primarily through the lens of slow genomic transcription. But the brain rapidly synthesizes endogenous neurosteroids, which are chemically modified analogs of hormones like progesterone, right there in the tissue.
And what do?
These neurosteroids are highly lipophilic. They accumulate in the neuronal membrane and can interact directly with the GABA A receptor. Functionally, they act somewhat remarkably like barbiturates, allosterically increasing the duration of the chloride channel opening and dramatically enhancing inhibitory tone.
So the natural fluctuations of these neurosteroids directly dictate our baseline levels of anxiety and mood.
Yes, and the most striking clinical application of this mechanism occurred recently with the FDA approval of brexanolone. Brexanolone is essentially a synthetic exogenous formulation of the naturally occurring neurosteroid allopregnanolone. It is administered as a continuous intravenous infusion specifically for the treatment of severe postpartum depression.
After childbirth, a woman experiences a massive precipitous crash in progesterone levels and consequently a crash in allopregnanolone. Her brain essentially loses a massive chunk of its natural braking system overnight.
Exactly. Brexanolone is a completely novel mechanism in psychiatry. Instead of waiting weeks for an SSRI to alter transcription, we are rapidly restoring the inhibitory GABAergic tone in a circuit that has been violently thrown out of balance by an endocrine crash.
Incredible. Just to round out the picture, we need to touch on the slow brakes. GABA receptors.
GABA receptors are entirely distinct structures. They are not ion channels. They are G-protein-coupled receptors, much like the mGluRs we discussed for glutamate. Uniquely, they function as obligate heterodimers. Two large 7-transmembrane units, GABA1 and GABA2, must bind together to function.
And what happens when they bind?
Postsynaptically, when activated, they cause a slow, long-lasting hyperpolarization by opening potassium channels, allowing positively charged potassium to slowly leak out of the cell. Presynaptically, they act as autoreceptors, inhibiting voltage-gated calcium channels to choke off further neurotransmitter release.
These are the targets for the muscle relaxant baclofen, but they are also the primary target for GHB gamma hydroxybutyrate. GHB has a very dark reputation as an illicit date rape drug because it rapidly induces profound, unarousable sleep and dense amnesia. And studies confirm it exerts this massive sedative effect by powerfully activating these slow GABA receptors.
Which brings us to the final and perhaps most vital part of our journey today. We have spent quite a long time rigorously dissecting the nuts and bolts of the accelerator and the brakes. We understand the locks, the keys, and the hazmat teams. Now we must integrate this. What happens when we apply this complex, mechanistic understanding to the reality of severe psychiatric illness? This is where the historical dogmas are completely falling apart.
Let's start with the big one, schizophrenia. For over half a century, if you asked any medical student what causes schizophrenia, the immediate reflexive answer was too much dopamine. And it makes sense, right? Every single antipsychotic drug ever brought to market works, at least in part, by blocking dopamine D2 receptors.
It is the reigning dogma. But as the Kaplan and Sadok text highlights, after 60 years of relying on the dopamine hypothesis, over two-thirds of treated patients remain substantially disabled. Why? Because the dopamine hypothesis is an incomplete downstream picture.
It doesn't tell a whole story.
Blocking dopamine treats the positive symptoms, the hallucinations and delusions, but it does absolutely nothing for, and sometimes worsens, the negative symptoms like flat affect and social withdrawal, or the profound cognitive deficits that truly disable the patient.
So if dopamine is just the smoke, what is the actual fire?
The field is rapidly coalescing around a much more fundamental circuit-level model driven by the interaction of glutamate and GABA. Post-mortem studies of brains from patients with schizophrenia consistently reveal a highly specific deficit. They show reduced expression of the GAD67 enzyme, specifically within the fast-spiking parvalbumin-positive GABAergic interneurons in the prefrontal cortex.
Those are the chandelier cells, the critical bottlenecks we discussed earlier that hold the massive excitatory pyramidal cells in check. So the brakes are failing. But why are they failing? Is it a primary defect in the GABA cells themselves?
The evidence strongly points to the primary defect actually being glutamatergic.
Yeah.
The leading circuit theory is the NMDA receptor hypofunction hypothesis. The foundational observation for this came from the clinical effects of dissociative anesthetics like ketamine and PCP.
Ketamine blocks the NMDA receptor pore, doesn't it?
Yes, these drugs are highly selective antagonists. When administered to healthy individuals, they induce a transient state that is clinically indistinguishable from schizophrenia. It doesn't just cause hallucinations, it perfectly mimics the negative affect and the cognitive disorganization.
Wait, let me get this straight. If you block an excitatory receptor, how does that lead to the hyperactive dopamine release that causes psychosis? That seems totally backward. Walk us through the exact circuit mechanics.
Let us trace the pathway step by step. The parvalbumin-positive GABAergic interneurons rely heavily on NMDA receptors to receive their activation signals. In schizophrenia, it is hypothesized that these specific NMDA receptors are hypofunctional. They are weak, faulty, or poorly expressed.
So the interneuron isn't getting the signal to fire.
Because they are hypofunctional, the GABAergic interneuron fails to activate properly. Because it fails to activate, it fails to release GABA.
And because it's a chandelier cell, its failure removes the brakes from thousands of glutamatergic pyramidal cells.
Exactly. The failure of the local brakes leads to profound disinhibition of the massive pyramidal cells. These excitatory cells lose their leash and begin firing chaotically and continuously. This uncontrolled, noisy glutamatergic static flooding the prefrontal cortex accounts directly for the cognitive deficits and the negative symptoms. The cortex is overwhelmed by computational noise.
But those pyramidal cells don't just stay in the cortex. They project long axons downwards into the subcortical regions, right into the midbrain where the dopamine is produced.
Precisely. This torrential, disinhibited wave of cortical glutamate crashes down onto the dopaminergic pathways in the striatum and midbrain. The glutamate heavily stimulates the dopamine cells, causing them to dump massive pathological amounts of dopamine into the system.
Yeah. So the excess subcortical dopamine, which causes the hallucinations and delusions, is therefore a secondary downstream consequence of a primary failure at the cortical NMDA synapse.
You just got it. The psychosis is just the engine revving out of control because the brake line was cut further up the chain.
Yeah. And modern genetics is finally providing the hard evidence to back up this circuit model, isn't it?
The advent of massive genome-wide association studies, or GWS, has been revolutionary. Researchers have analyzed the genomes of hundreds of thousands of subjects and identified over 100 specific genetic risk loci for schizophrenia. And when you map these genes to their biological functions, the pattern is undeniable. the genetic risk converges heavily on the structural and functional integrity of the glutamatergic synapse.
What kind of genes are we talking about?
We see risk genes encoding voltage-dependent calcium channels, AMPA receptor subunits, scaffolding proteins, and critically, the SRR gene.
The SRR gene, that codes for serine racemase. The enzyme we talked about earlier that the brain uses to convert L-serine into D-serine, which is the necessary key for the glycine modulatory site on the NMDA receptor.
Yes. If you engineer a mouse that genetically lacks serine racemase, you eliminate its ability to produce D-serine in the forebrain. The NMDA receptors lose their co-agonist and become chronically hypofunctional. That mouse develops a phenotype strikingly similar to human schizophrenia. A physical loss of dendritic spines, severe cognitive deficits, and elevated striatal dopamine release.
That is profound.
Furthermore, the GWA's data strongly highlights variations in component C4, a protein involved in the immune system that also mediates the physical pruning of excitatory synapses during late adolescence.
Which perfectly aligns with why schizophrenia typically presents in late adolescence or early adulthood. The immune system aggressively overprunes an already weakened glutamatergic network until the whole circuit just destabilizes. It is a profound structural rethinking of the illness. But what about depression and anxiety? We usually think of serotonin for depression.
Serotonin remains an important modulator, but the core machinery again relies on amino acids. MRS neuroimaging reveals significant reductions in cortical GABA levels in patients with major depressive disorder and panic disorder. When the depression is effectively treated and the patient reaches remission, whether by SSRIs, cognitive therapy, or electroconvulsive therapy, those cortical GABA levels are shown to normalize.
But the most dramatic, paradigm-breaking shift in our understanding of depression has come, once again, from manipulating the NMDA receptor.
You're talking about the ketamine revolution.
I am. For 70 years, the dogma stated that antidepressants like SSRIs take 4 to 6 weeks to alter gene transcription and produce a clinical effect. But clinical trials demonstrated that a single sub-anesthetic intravenous infusion of ketamine, which, remember, is an NMDA receptor antagonist, can produce a robust, substantial reduction in depressive symptoms and a near-instantaneous cessation of severe suicidal ideation, often within hours.
In 2019, this culminated in the FDA approving esketamine, a nasal spray formulation, for treatment-resistant depression.
But again, this presents a mechanistic paradox. Ketamine blocks the NMDA receptor. It blocks the primary mechanism for learning and plasticity. How does blocking plasticity cure depression overnight?
It's an area of intense ongoing research, but the leading consensus relies on the same microcircuit logic we discussed for schizophrenia. Ketamine is thought to preferentially block the NMDA receptors located on those highly active local GABAergic interneurons.
So it temporarily cuts the brake.
Yes. This transient blockade triggers a rapid, controlled burst of glutamate release from the disinhibited pyramidal cells. But because the NMDA receptors are blocked by the ketamine, this glutamate surge bypasses them and heavily activates the remaining AMPA receptors.
And what does that intense AMPA activation do?
It triggers a massive intracellular release of brain-derived neurotrophic factor, or BDNF.
BDNF is essentially molecular miracle-grow for the brain.
Exactly. The BDNF rapidly stimulates the growth of new dendritic spines and physical synaptic connections. Chronic stress and severe depression are known to cause the physical atrophy and withering of apical dendrites in the prefrontal cortex. Ketamine forces a rapid structural regrowth of those withered circuits. It chemically forces the brain to repair its own hardware.
We are mechanically rebuilding the circuits of hope in a matter of hours. It's breathtaking. Let's look at one final, very common clinical scenario before we wrap up. Alcoholism. Alcohol use disorder is perhaps the most widespread, tragic example of a chemical violently manipulating this exact excitation-inhibition balance on a daily basis.
Acute alcohol intoxication acts as a sledgehammer to the delicate tone of the metropolis. Ethanol simultaneously alters both systems. It significantly enhances the function of the GABA-A receptors, augmenting the brakes, producing sedation, slurred speech, and anxiolysis. Simultaneously, it acts as an uncompetitive antagonist at the NMDA receptors, cutting the accelerator, profoundly impairing memory formation and coordination.
But the brain is a homeostatic machine. It fights back. It doesn't like being pushed out of its baseline tone. If a person drinks heavily and chronically, the brain has to adapt to this continuous state of sedation, right?
It adapts aggressively to survive. To counter the chronic chemical suppression, the brain fundamentally alters its transcription. It physically downregulates or removes GABA receptors from the synapses
Hmm.
and massively upregulates or builds more NMDA receptors.
It's trying to wake itself up.
The brain is desperately increasing its excitatory drive, trying to stay awake and functional despite the continuous presence of a depressant. This physical restructuring of the receptors is the biological basis of clinical tolerance. You physically need more alcohol to achieve the same effect because the underlying hardware has become less sensitive to it.
And this brilliantly explains the absolute terror of severe alcohol withdrawal. If a chronic alcoholic suddenly stops drinking, the ethanol clears their system in a matter of hours. But the hardware changes. The decimated GABA breaks, and the massive, overgrown excess of NMDA accelerators remain in place.
Without the heavy dampening effect of the alcohol holding the system down, the brain is suddenly left with a violently hyperexcitable architecture. The massive excess of NMDA receptors is suddenly exposed to normal baseline glutamate, resulting in overwhelming, unregulated excitation.
Clinically, this manifests as severe tremors, hallucinations, seizures, and the life-threatening hyperautonomic state of delirium tremens,
The brain is effectively burning itself out through massive excitotoxicity.
which explains how a specific pharmacological treatment works. A camprosate is a drug used to maintain abstinence in recovering alcoholics. It doesn't act like a traditional sedative. It is thought to structurally modulate the NMDA receptor to gently reduce this chronic hyperglutamatergic tone, easing the physical cravings and stabilizing the over-revved recovering brain. But to truly understand the sheer power of NMDA, we have to look at what happens when alcohol is introduced not to an adult, but to a fetus.
Fetal alcohol syndrome highlights a profound developmental paradox that perfectly underscores everything we have discussed today. We established earlier that in an adult brain, overactivation of NMDA causes excitotoxic death, and blocking it protects the brain. But in a developing fetal brain, the rules are inverted.
Why?
Because during embryogenesis and fetal development, migrating neurons require constant rhythmic pulses of calcium through the NMDA receptor simply to survive. It signals to the cell that it has successfully made a synaptic connection. It is a survival signal.
So if ethanol crosses the placenta,
If it crosses the placenta and blocks those fetal NMDA receptors, the developing neurons are deprived of that essential calcium signal. Thinking they have failed to connect, they initiate widespread apoptosis programmed cell death. Millions of developing neurons commit suicide simply because the accelerator pedals was temporarily blocked. This results in the tragic, irreversible microcephaly and intellectual disability characteristic of fetal alcohol syndrome.
it perfectly illustrates our central thesis. Whether it is a fetus building a healthy cortex, a student learning a new language, a patient sliding into the disorganized noise of psychosis, or an addict recovering from withdrawal, the core, fundamental mechanism remains the same. It is the continuous, dynamic negotiation between the excitatory drive of glutamate and the inhibitory restraint of GABA.
The era of categorizing psychiatric illnesses purely by symptom checklists is coming to an end. As our genomic and neuroimaging tools become more precise, we will transition from prescribing broad chemical blankets to targeting specific, measurable aberrations in synaptic physiology.
What if in the near future we don't diagnose you with depression or anxiety, but rather with a specific tuning error in a specific microcircuit's excitation inhibition balance? What if the future of psychiatry looks less like blunt pharmacology and more like precisely tuning a grand piano? That is the paradigm shift Kaplan and Sadok are preparing us for.
It is a profound shift to consider.
Before we go, let's test your mental models with some clinical reasoning questions based directly on what we just covered. This is the best way to solidify the mechanisms we just explored. Take a second after I read the question to think of the answer.
This is always the best part.
Question 1. You are tracing the metabolic pathway of neurotransmitters. Which specific cell type is exclusively responsible for expressing glutamine synthetase to safely recycle synaptic glutamate?
Think about the hazmat team.
Answer. Astrocytes. They use glutamine synthetase to convert volatile glutamate into safe, inert glutamine for transport.
Spot on.
Question 2. The NMDA receptor is known as a coincidence detector. Besides glutamate binding to the NR2 subunit and sufficient membrane depolarization to remove the magnesium plug, what third factor is absolutely required for it to open in the forebrain?
Second key.
Answer. The binding of D-serine to the glycine modulatory site on the NR1 subunit. Question 3. A patient is administered a high dose of barbiturate and suffers respiratory arrest. How does the mechanism of barbiturates differ from benzodiazepines at the GABA receptor to make them so lethal in overdose?
Think of the club owner versus the manager.
Answer. While benzodiazepines only increase the frequency of channel opening in the presence of natural GABA, barbiturates increase the duration of the open state and, crucially, can directly open the chloride channel themselves at high doses, independent of GABA.
Which is incredibly dangerous.
Question 4. Based on the NMDA receptor hypofunction hypothesis of schizophrenia, what is the primary downstream effect when NMDA receptors fail on parvalbumin-positive GABAergic interneurons?
The chandelier cells failing.
Answer. Disinhibition. The failure of the local brakes allows pyramidal neurons to fire excessively, leading to cortical noise and secondary excessive subcortical dopamine release.
Go on again.
Question 5. A patient with Alzheimer's disease is prescribed memantine. What is the precise mechanism by which beta-amyloid fibrils contribute to excitotoxicity, necessitating this drug?
The slow leak.
Answer. The fibrils chronically, mildly depolarize the neuronal membranes. This constant depolarization removes the protective magnesium block from NMDA receptors, allowing a constant toxic background leak of calcium.
Excellent.
Question 6. You are reviewing novel treatments for severe PTSD. Which partial agonist at the NMDA receptor has been shown to chemically prime the brain to enhance the active learning required for fear extinction during psychotherapy?
The old tuberculosis drugs.
PSYCH.
Brilliant.
Here are the details of this episode.
Amino acid neurotransmitters. Byline. The brain's primary dialects excitation and inhibition in constant negotiation. Episode description. At the heart of neural communication lie a small group of powerful molecules that set the tone of brain activity. This chapter focuses on amino acid neurotransmitters, particularly glutamate and GABA, which together form the fundamental balance between excitation and inhibition. In this episode, we explore how glutamate drives neural activation, enabling signaling, plasticity, and learning, while GABA provides restraint, stabilizing circuits, and preventing excessive activity. The brain depends on this delicate equilibrium. Too much excitation risks instability, too much inhibition risks suppression. We examine how these neurotransmitters act through different receptor systems, shaping both rapid signaling and longer-term modulation. Their influence extends across virtually all brain systems, making them central to both normal function and pathology. Disruptions in this balance are implicated in a wide range of psychiatric conditions from anxiety and epilepsy to schizophrenia and mood disorders. Rather than isolated dysfunctions, these represent shifts in the overall tone of neural networks. This chapter reframes brain activity as a dynamic negotiation, a continuous balancing act that allows complexity without chaos. Key takeaways. Amino acid neurotransmitters, primarily glutamate and GABA, are central to brain function. Glutamate is the main excitatory neurotransmitter. GABA is the main inhibitory neurotransmitter. Brain function depends on a precise balance between excitation and inhibition. Different receptor types mediate fast and slow signaling effects. These systems are widely distributed and influence most neural circuits. Dysregulation of excitation-inhibition balance is implicated in multiple psychiatric disorders. Understanding this balance is key to interpreting both symptoms and treatments.